All right, so we're back. And here we have our mitochondria. And this is very similar to the model or the diagram that you have in your journal as well, or in your notes from the copy of the notes that I gave you either through Google Classroom or in person. And what you've got here is the mitochondria, right? We've labeled that at the top. I've also included the equation for cellular respiration that you should be familiar with from 7th grade, although I get that that was a couple years ago, so we might need a reminder on that. And we'll talk about it here in just a second. You also have some information near the top that talks about glucose, as well as some ATP that's involved in creating pyruvate. We'll talk about that here in just a second. You've got this interesting little cycle here with some arrows shooting off of it called the Krebs cycle. And then you have these little boxes with some circles and these little darker boxes that are part of this inner membrane, if you will. So let's talk about the structure of this so that we can understand how cellular respiration is going to occur within the mitochondria. I'm going to keep my notes handy just to make sure that I don't forget anything that I've already mentioned, to make sure that we're all good with this information. All right, so the first thing we're going to focus on here is the actual structure, the overall structure of the mitochondria. And we're going to zoom in on two sections up here near the top. I know there's a little bit of a glare down here, and I do apologize for how dark the video is. I'm trying to make sure you can see everything on the board without too much glare, but we won't be using this space, so it should be okay. All right, so over here at the top, we're going to focus or zoom in a little bit on both of these sections. These are going to represent the membrane, a very specific type of membrane known as a phospholipid bilayer. Now, this should sound familiar to you, as we have already talked about phospholipid bilayers when we looked at the cell membrane, the membrane that surrounds the entire cell. And we did say that it was a phospholipid bilayer. So we have that phosphate head with the fatty acid tails that are facing each other, right? Creating this double layer, if you will, of phospholipids. Now, the mitochondria is unique in that it has an outer phospholipid bilayer that we actually call the cristae. And you have a word bank on your notes that will include a lot of these terms in case you can't quite make out the spelling. Hopefully my handwriting's neat enough for you to see this. So you have the outer membrane known as the cristae, which is a phospholipid bilayer. But notice I zoomed in on this inner membrane and said that it is also a phospholipid bilayer. And that is why we will refer to the mitochondria as having and I already made a mistake. Let me fix that for us. A double phospholipid bilayer. So one phospholipid bilayer on the outside called the cristae, and then the inner membrane is also a phospholipid bilayer. So 4 layers of phospholipids essentially. Now, in terms of what do we care about regarding these two membranes? Well, obviously the structure itself is unique, but more importantly, the inner membrane is the membrane that has a lot of folds in it. It's one way that we've described the mitochondria as having an inner membrane with lots of folds. And the reason for those folds is to increase surface area. Now, the inner membrane is the site where ATP is going to be produced. If we can create more surface area on the inside, on that inner membrane, we have more sites, more places for ATP to be synthesized, which means we can make more energy. All right, so hence the folded membrane on the inside. Now, I've kind of minimized the amount of folds in there just to create some space so we can fill out the information we need to know regarding the synthesis of ATP. So keep in mind, I've really minimized the folds here. Should be a lot more folded than this. All right, so moving forward, we have space in between these two membranes that we're going to refer to as the intermembrane space. I'm going to abbreviate it IMS for short. All right, so the intermembrane space is the space between the inner membrane and the outer membrane. The word itself kind of defines itself, inter meaning between, and then membrane space. So the space in between the two phospholipid bilayers is the intermembrane space. Very important spot for us, so we'll get to that here in just a second. The space on the inside of the mitochondria, just outside of the inner membrane, is going to be referred to as the matrix. And we need to know those locations because they're going to play a pretty important role in making sure that we can actually create ATP. All right, so how do we actually make ATP? Well, we have what we know as a molecular motor. That's this little box right here with the 3 circles on it. This is a molecular motor that we're going to refer to as ATP synthase. ATP synthase. Now, if you recall from our lesson on enzymes, words that end in ASE are typically going to be reserved for enzymes. So this is an enzyme, which by the way is a molecular motor, okay, which is important because it's going to make sense here in just a second. But it's an enzyme that synthesizes or makes ATP. All right, so once again, the name itself kind of gives away the meaning here. So it's an enzyme that synthesizes or makes ATP. That's these little boxes here on either side with the three little circles. And keep in mind, these are going to be located all along the inner membrane. This is why we want to increase surface area so we can have a lot of these molecular motors synthesizing ATP for us. All right, now, two qualities of ATP synthase that are really important and are going to help us understand how we create energy in the form of ATP. The first of which is that ATP synthase is hollow. It's hollow, all right? It's kind of like a tube, a cylinder or tunnel, if you will. And two, it spins. All right, these are two qualities of ATP synthase that allow it to make ATP. Now, if you recall from previously, when we looked at the molecule of ATP and what it was made out of, what we had was a molecule of adenosine. Remember, adenosine was the adenine and that carbohydrate ribose combined made adenosine. And adenosine was bonded to 3 phosphates. 3 phosphates. Remember, phosphate is PO4. All right. So adenosine triphosphate is the molecule of ATP that we're going to use for energy. The way we get energy from this, if you recall, is that we break the bonds holding the atoms together. When we break those bonds, the energy that holds them in together is going to be released into energy that we can use. This is going to occur in any chemical reaction where energy is released. We break bonds to release energy. Now, the goal in regards to using ATP for energy is to break 1 phosphate off of the molecule. So if we can break 1 phosphate off the molecule, we release the energy that our cells need to function properly. Now, what that's going to leave us with when we use ATP and we break those bonds, it's going to leave us with this 1 phosphate right? So we have 1 phosphate right here all by itself now because we broke that bond. And all the rest of this molecule right here is now going to be known as ADP, right? ADP is adenosine diphosphate, di meaning 2, right? It's a prefix we should be familiar with for 2, all right? So we have adenosine diphosphate and a single phosphate. When we break the bond holding that phosphate to the rest of the molecule, we release the energy, and now we have a molecule called ADP and a phosphate molecule all on its own. The goal of ATP synthase is to make ATP from these materials. All the ATP that we have that we're going to use within our cells is going to be recycled. And so here's what happens. Here's an ATP molecule, for example. I've broken the bond that holds this phosphate. We'll call this a phosphate, right? So here's my phosphate. Here's ADP. Here's that top part here. And now they're kind of floating around out here. I've already used their energy. Now I want to make ATP. Now, one way to make ATP is to take this phosphate and combine it with ADP, which is what I had before. That's going to make adenosine triphosphate, ATP. However, if you recall from the previous video or from the previous section, if I put these all together into one video, this has a phosphate that's negative and this has a phosphate that's negative. Those oxygens are negative, which means they are not normally or naturally going to want to bond together if I can not drop it, right? They don't want to bond together. They're negatively charged, so they're going to repel each other. On their own, they will not bond. So what we have to do is you have to force them to bond. We need to get them close enough so that the covalent bond that forms between these phosphates can hold them together despite that negative charge trying to repel them. Because that's what's happening with the rest of the phosphates anyway. Remember, it's unstable, but it is held together by those covalent bonds. So we just need to kind of force that bond. And once we do, it'll stay bonded to create ATP. Then we can break the bond to release the energy, use it again, and then force them back together to make ATP. And we keep doing this with all of our adenosine diphosphates and all of our phosphates. We just put them back together to make ATP, break them apart to release the energy, put them back together, and we recycle these ATP molecules. ATP synthase, this is the molecular motor. This is the enzyme that's helping us put these back together. And the way they do that, one, they're hollow, and two, they spin. So let's talk about how this works. ATP synthase, remember these don't want to attract to each other because they're negatively charged. ATP synthase is going to grab hold of one, it's going to grab hold of the other, and it's going to spin and force those bonds. Now, it can't just bring them into the same area and wait for them to bond on their own. Remember, they're going to repel. So ATP synthase, because it spins, is able to generate, let's say, for example, it's not quite the same as physics, but let's just say, to help us understand this better, that we have to generate a certain amount of force, so that spinning helps us force those molecules together, right? So this is the benefit of ATP synthase spinning. It's able to force that phosphate onto ADP and make them bond, creating ATP. Now, because this is a motor, molecular motor, something needs to power it. We need an energy source. Now, ideally, we don't want to use ATP to run an ATP making machine because we're going to net, well, not much. We're either going to break even or not make as much energy as we're hoping to get from ATP. So we don't want to use ATP to power this process. But one of the benefits of ATP synthase being hollow is that if protons, here's some protons, and we know protons are positively charged, so I'm going to use little pluses to represent protons. If protons pass through ATP synthase from the IMS to the matrix, if ATP synthase has protons going through it from the inside to the outside, it's going to make ATP synthase spin. And we want it to spin because spinning takes that phosphate and that ADP and slams them together to create ATP, right? So we want it to spin. We're going to make it spin by having protons go through it from the IMS to the matrix, all right? Now, what's going to make these protons go through ATP synthase is mainly the fact that it's hollow, but more importantly, facilitated diffusion. And when you recall from transport, facilitated diffusion does not require energy because it's passive transport. Now we got to go back and go, all right, well, how do we make this diffuse through ATP synthase? Well, the way diffusion works is you need a higher concentration of particles on one side of the membrane and a lower concentration of particles on the other side of the membrane. So if we have more protons on the inside, they are going to move from an area of high concentration to an area of low concentration on the outside. That's diffusion. Why is it facilitated? These protons cannot pass through this phospholipid bilayer on their own. And so ATP synthase, because it is hollow, is going to provide that channel or that tunnel or that opening for protons to go through. They want to go from areas of high concentration to areas of low concentration. Also, they're all positively charged particles, which means they're repelling each other anyway and kind of fighting for space. This opening gives them that opportunity to move away from these other positively charged particles. However, this brings us to our first problem. If we think back to diffusion, we know that essentially what we're going to end up with is a moment where we have an equal number of molecules on both sides of the membrane. When that happens, we call it equilibrium. And for the most part, when we look at cell function, we want equilibrium, we want homeostasis, right? We want things to be working in balance. However, in this instance, equilibrium is going to be a bad thing for us. Why is it a bad thing? Well, if I have equal number of protons on the inside and an equal number of protons on the outside, I'm not moving from an area of high concentration to an area of low concentration, which means any protons that go through ATP synthase, like through the IMS to the matrix, are also going to be moving back from the matrix towards the IMS. So I'm going to have movement. It's not going to stop flowing, but I'm going to have movement going in both directions now. And anytime a proton goes through, another one's going to come back, and it's essentially going to kind of cancel out. It's going to spin the wrong way when it's going the wrong way, and I'm not going to be able to slam those phosphates into ADP and create ATP. If I can't make this spin properly, I can't make ATP, and then I die. We want to avoid that, so we need to figure out a way to keep the concentration of protons on the inside, on the IMS, higher than the concentration of protons in the matrix. So we have some other tools to help us do that. Along the inner membrane, we also have these dark boxes that are on your diagram as well, that are going to act as, here's a little arrow, might be hard for you to see, a little arrow. they're going to act as proton pumps, which literally does what it sounds like it does. It takes protons and it pumps them back into the IMS. These are our proton pumps. So they're going to take protons in the matrix and they're going to pump them back into the IMS. So long as these are continuing to function properly, and all of them are, and it's mirrored on both sides of this fold here. So anything happening on this side is happening on the other side, in case that's confusing anyone. These three pumps here are going to be pumping protons back into the IMS to keep the concentration higher on the inside so that we can diffuse protons through ATP synthase so that ATP synthase can spin, so that we can force our phosphates onto our ADP and create ATP so that we have the energy for our cells to stay alive and for us to stay alive. All right. So proton pumps are pumping protons back into the IMS so that we can make ATP using ATP synthase. Now, if you recall, anytime you see the word pump, we know that's a form of active transport. Active transport requires energy. And as I mentioned before, we don't want to use ATP to run an ATP making machine, because then we're going to net fewer ATP, or worse, break even. And if we're breaking even, we're not providing energy for anything other than the machine making ATP, and our cells can't do anything else, which means we die. We don't want that to happen. So our proton pumps need energy. And for this one, we're going to kind of cheat and go back to the beginning, even though we're starting at the end, because this is the end of ATP synthesis. And we're going to jump to how do we power our pumps? We need to go outside of the mitochondria for this. So we're going to go outside the mitochondria, and you're going to have on the top of your diagram this section that says glucose to pyruvate, and it says there's 2 ATP and 4 ATP. Now what this is showing is a process that we call glycolysis. All right, this is glycolysis. And this is another one of those key terms in your word bank. Glycolysis, if you will. That's what's happening over here. Now, if we break that word down, glycolysis, lysis means to split or to burst. We use this word when we're referring to the process that viruses use to replicate. The lytic cycle will cause a cell to burst or lyse. So lysis means to split or break. Glyco is the prefix or root that means glucose. So we are splitting a glucose molecule. Now, glucose being the carbohydrate that we like to use for energy, right? So glucose gets split, and it takes a little bit of energy to do that, 2 ATP, and then we get 4 ATP from glycolysis, which means we net 2. It costs 2 to get 4, so we really only ended up with an extra 2. So not a lot of energy produced from that. And that'll create 2 pyruvates. So if we split glucose, we end up with 2 pyruvates, which means pyruvate is half a glucose. Right. Now, when we do that, pyruvate will find its way into the mitochondria into what is known as the Krebs cycle. We're not going to go too in depth with the Krebs cycle. We just need to know a couple of things about it, right? In the Krebs cycle, we have NAD plus and FAD. Now, NAD plus and FAD are what are known as electron carriers. They're really good at picking up electrons. So when we take glucose and we split it into 2 pyruvates, essentially what we're doing is we're taking this C6H12O6, which is glucose, and splitting it in half. So originally, and we're going to follow the carbons here because I think it makes sense if we just pick one element here. If we follow the carbons, now pyruvate, each one, because there's 2, have 3 carbons. Now carbons for C3 are going to look like this. So here I have 1 carbon, 2 carbons, 3 carbons. Now I don't need this cap. You're going to see why here in a second. And these carbons are going to be held together by the bonds that hold any molecule together. And those are going to be because of the electrons. The valence electrons are the ones responsible for bonding. So I have electrons that are holding my 3 carbons together. When pyruvate enters the Krebs cycle, NAD plus and FAD, being electron carriers, are really good at picking up electrons. Or another way to look at it would be they're really good at stealing electrons. So what they're going to do for this carbon, and we're just focusing on carbon right now because we want to make this simple, is they're going to come in and they're going to take the electrons that hold carbon together. Our C3 is now 3 separate carbons because our FAD and our NAD plus stole electrons from it. They took the electrons that are holding them together, so now I end up with 3 separate carbons and a handful of electrons. Now, why do I want electrons? Well, the reason I want electrons is because if you go back to what you remember from chemistry, now this is not exactly the same, but it's a good analogy. When you have, and I'm not going to ask you to add this to your diagram, I just don't have anywhere else to put it. When you have an atom, here's our nucleus, you're going to have these electron shells or energy levels that surround the nucleus. The electrons are going to be hanging out up here. 2 within the first shell, and then up to 8 in the second shell or energy level. When you add energy to an atom, for example, when you take something that's dark in color and you put it outside and the sunlight hits it, that sunlight energy is going to cause the electrons to move up a level. They're going to go up to the next level because they've absorbed that energy. The electrons are going to move to a new energy level. When the electrons go back to their original place, when these drop back down, they release the energy they absorbed, which is why when you touch something that's like, say, black in color, that's been outside absorbing sunlight energy, when you touch it, it's going to be warm. It's going to release the energy in the form of heat. And when it releases that energy, the electrons go back down to their original spot. So one way to look at what's going to happen here in a second is that when electrons go down a level, they release energy. Once again, it's not exactly like that, but it does help us hopefully understand the process here. Okay, so when electrons move down a level, they release energy. So here's what's going to happen. When NAD plus and FAD pick up electrons, right, because they're really good at stealing electrons, they become NADH and FADH2. So NAD plus, FAD, those are shorter than what we just wrote, NADH and FADH2, because they didn't have electrons. Now that they have them, they become bigger. One way to help you remember which one's which. All right, so when NADH and FADH2 have their electrons, what they're going to do is NADH is going to drop off electrons at pump 1. We're going to call that top one pump 1. FADH2 is going to drop off electrons at pump 2. NADH has more electrons than FADH2 does, so it drops off at the first pump, and it's going to power all 3 pumps. FADH2 has a few less electrons, so it's going to drop off at pump 2 and power pump 2 and pump 3. Now here's what's going to happen. NADH drops off electrons at pump 1. Those electrons get transported to pump 2. Those electrons then get transported to pump 3. And every time these electrons move down a level, every time they move down a level, like that model of the atom I had up here, they're going to release energy. And that is the energy that's going to power our proton pumps. Proton pumps are pumping protons back into the IMS so that we have a higher concentration, so that those protons will diffuse through ATP synthase because it's hollow. It will make it spin, and when it spins, it forces a phosphate onto ADP, brings it around, spins it over, slams those two together to create that bond so we can have ATP molecules. All right? Now, because these proton pumps are being powered by electrons, we're going to call this whole system right here, not ATP synthase, but our 3 pumps and the transport proteins in between, the ETC. And in your notes, it will say the electron transport chain, okay? Because that's what it is. It's a chain of pumps that are transporting electrons from pump 1 to pump 2 to pump 3. And every time it moves down a level, it releases the energy that's going to power the pump to get those protons back into the IMS. All right. Now, all of that came from food. We eat food so that we can steal electrons from it, which is one way of looking at what food is. Food is anything you can steal electrons from, which is why we can't eat certain substances. We can't eat dirt, we can't eat plastic, because we cannot break it down and steal electrons from it. Glucose is definitely the preferred method here. Now, if we don't have enough glucose, then yes, we can use our triglycerides, our fatty acids, our lipids, if you will, for energy storage. So if you stop eating food, this process doesn't stop working. You just start to use your own fat stores or even your muscle tissue if you deplete your fat stores. Now, obviously, this can't go on too long because you'll run out of resources to steal electrons from. And if you run out of electrons, you can't power the pumps, which means you can't get protons back in the IMS, which means the concentration drops. ATP synthase won't spin because protons are not going through it anymore. You can't make ATP, and then you die. And this is what starving to death would be. We have nothing to steal electrons from to power our pumps. Now, when you take this information, you start looking at the formula that's set up for this, and we're going to recap this whole thing here in just a second. You're going to notice that we have something that we have not talked about yet, and that is we have glucose, we need oxygen, right? We're going to produce carbon dioxide, water, and energy. Now I put up there 30 plus ATP. This process will create essentially 30 to 38 ATP molecules. So it's very effective. Glycolysis produces 4, which technically is just 2 because it costs us 2. So this is not a preferred method of ATP production. This is much better. So if you have mitochondria, you can make a lot of ATP. But we got to think about, why is this oxygen here? Remember, this is called cellular respiration. Well, at some point, as electrons are moving down from pump 1 to pump 2 to pump 3 is the last stop. And at some point, I'm going to start dumping off a lot of electrons into pump 3, which is negatively charged because electrons are negatively charged, which means at some point, it's going to get full and it's going to start to repel electrons from pump 2. Even with energy pushing those down the chain, at some point, pump 3 is going to have so many electrons, it's going to repel pump 2 from sending any more electrons to it, which means pump 2 is going to get full and won't receive electrons from pump 1. And if I can't move electrons from pump 1 to pump 2 to pump 3, if electrons cannot go down a level, they can't release their energy, I can't power my pumps, I can't create a higher concentration on the IMS, ATP synthase is not going to spin, I can't make ATP, and I die. So we need a way to get electrons away from pump 3. We need something to come in and take electrons away from pump 3. And one of the best things to use is oxygen. Now, oxygen on its own, once again, don't draw this on your diagram, I just don't have anywhere else to do it. Oxygen on its own has 6 valence electrons, which means it needs 2 more to be stable. If you remember the octet rule, 8 electrons in your valence shell is stable. So it's going to bond with oxygen. Oxygen is really good at bonding with oxygen. They just share 2 electrons each, and now they have 8 each, and they're stable. And this is O2. This is the oxygen we breathe. This is O2, right? So O2 is 2 oxygens that are covalently bonded. They're sharing electrons. Now, I'm not sure it exactly works like this, but here's how I like to look at this. If given the option of sharing electrons with someone else or finding your own, the preferred method would be to find your own. And we happen to have a lot of electrons that we need to move down here in pump 3. So what oxygen is going to do, we're going to breathe in oxygen, right? We breathe in oxygen. Oxygen is going to move through our mitochondria down here to pump 3. And here's O2. It's going to come down to pump 3. It's going to pick up some electrons, and it's going to turn into 2 water molecules. And don't worry, I'm going to explain how it turns into water here in just a second. So oxygen is going to swoop down here to pump 3, pick up some electrons, and then leave as water. And here's how this works. So right now they're sharing electrons. Well, they don't want to share, they want their own. So they're going to stop sharing. In the presence of more electrons, they're going to pick up 2 each. So here's my electrons that I picked up. These, I didn't have these to begin with. So I have 2 oxygens now that each picked up 2 extra electrons. Now here's what's going to happen. And this is some stuff you're going to learn more about in chemistry next year, but I want you to understand how this turns into water. Originally, oxygen only has 8 total electrons. It has 2 in its first shell, 6 in its outer shell. 8 total electrons. It also has 8 protons. This means that all my protons that are positive and all my electrons that are negative cancel each other out. And oxygen is neutral. 8 protons, 8 electrons, 8 pluses, 8 minuses, they all cancel out and you're neutral. But now we've picked up 2 extra electrons. So it's going to be charged by those electrons. Excuse me. It's going to have a charge of -2. Because it picked up 2 extra negatives, it has a charge of -2. Now, what we know about negative charges is that they attract positive charges. So these negative oxygens are going to be attracting positive protons. And it just so happens that we've been pumping protons out into the IMS, right? We're pumping protons out of the IMS into the matrix where oxygen is picking up electrons from pump 3. So these -2 are going to attract 2 protons. If you take a proton and an electron, one of each, you take 1 proton and 1 electron and you put them together, you create hydrogen. If you remember the periodic table, hydrogen's up here in the top corner, hydrogen is 1 because it has 1 proton and 1 electron. Doesn't need a neutron to be hydrogen. So each of these oxygens that are now -2 attract two protons that are out here in the matrix, right? Pick those up, that proton electron becomes hydrogen and it picks up two. So now you have two hydrogen with one oxygen, which is H2O, which is water. And we make two of them because we had two oxygens that split to pick up these electrons. Now, that is essentially the only reason that we have to breathe oxygen. The entire respiratory system and cardiovascular system that helps transport oxygenated blood to different parts of the body, the whole point of respiration, remember cellular respiration, is so that we can breathe in oxygen to take electrons away from pump 3, which creates space for electrons to move down from pump 2, which creates space for electrons to move down from pump 1, and we can keep transporting electrons down the chain to release energy to power the pumps that bring protons back into the IMS, so the concentration is higher, so they will flow through ATP synthase, which makes it spin, which forces those phosphates to slam into those ADP molecules to create ATP. So we eat food to steal electrons, and we breathe oxygen to steal electrons from pump 3. All of this is about managing our electrons. And if we can manage our electrons properly, we can maintain the concentration of protons that we need on the IMS or in the IMS to create that facilitated diffusion passive transport that's going to make ATP synthase spin. Now I'm going to move this because this doesn't really belong here. And essentially that is cellular respiration. It does leave us with a little bit of information in our formula or an equation that we haven't covered yet. And that is essentially this. Here's our glucose. We use glycolysis to break it down into pyruvate to get the electrons from the Krebs cycle. We breathe in oxygen, there's our O2, so we can steal electrons from pump 3. That's going to produce 6 CO2, 6 H2O, and 30 plus ATP, between 30 and 38 ATP molecules. Now we know where the water comes from. The water is going to come from oxygen stealing electrons from the electron transport chain from pump 3 and picking up a couple protons to create water. This is water that we're going to exhale when we breathe out. CO2, we know that we breathe out CO2. We breathe in oxygen, we breathe out CO2, but where's that CO2 coming from? Because I don't see it in this process of ATP synthase. Well, the CO2 is going to come from the Krebs cycle. Like I said, we're not going to go into too much depth with the Krebs cycle, but keep in mind, pyruvate came in here, right? I had my 3 carbons, NADH and FADH2, picked up those electrons, right? Stole those electrons from the carbon molecule. And so I had 3 floating carbons just kind of floating around out there that are not stable either. They want some electrons. I'm still breathing in oxygen that is also not stable. It wants wants electrons as well. And so up here in the Krebs, we're actually going to have some CO2 produced. This is where CO2 is going to be produced. Those carbons that don't have their electrons holding them together anymore are going to join up with some of the oxygen that we're breathing in to create CO2. Carbon and oxygen bond together really well. They'll create those covalent bonds to share electrons. And this is where we're going to create the CO2 that we breathe out as well. All right. So it's going to happen in the Krebs cycle, which we're not going to spend too much time on. Now, all of this, known as cellular respiration, is, well, it's going to require respiration. It's in an oxygen-rich environment, which is why we're breathing as much as we do. This is known as aerobic respiration. Now, in the case that we have anaerobic respiration, in the event that we have low CO2, and this will be the last thing that we cover here. I'm sorry, low O2, not CO2, low oxygen environment. So we do glycolysis. We're trying to create energy, and glycolysis will create energy for us, just not as much as the mitochondria will. In the presence of very little oxygen, a very low O2 environment, we're not going to be able to go through this entire process very effectively or efficiently, because we're going to start to produce something called lactic acid. And if you do anything with athletics, if you're an athlete yourself, if you ever work out, then you understand that lactic acid buildup in the muscles is going to make the muscles a little bit harder to use. It's going to make you a little bit more sluggish. It's going to make the muscles work a little bit slower and may even cause a little bit of discomfort or pain. And this is why essentially the first push-up is easier than the 10th push-up. Because as we are working out, as our muscles are doing more than just keeping us alive, just being here, being alive requires energy. When we start to ask more of it, we require more energy for that. And so all of this process is creating ATP to help our cells keep up with the processes that keep us alive. And now we're asking it to do more. We're asking our body to do more. We're asking it to exert itself and use our muscles to a higher level. This requires more energy. Well, at some point, we're going to start to fall behind. We're going to be using more energy than we're producing because ATP doesn't last very long. It's got a half-life of about a minute. So because it doesn't last very long and we're burning up a lot of ATP and trying to keep up with making ATP, your body, your brain is pretty smart here. It wants to maintain balance. And so it's going to start to produce lactic acid in a low oxygen environment, which is going to make your muscles harder to use, which is going to essentially force you to slow down and eventually stop you from exerting yourself to the degree that you were. You're going to reach a point where you can't lift that weight anymore. You can't do another push-up or you can't run another lap. And when your body has built up enough lactic acid within the muscles that makes them want to stop working, you stop exerting all that energy, which gives your body a chance to catch up. You stop all of the extra that you're doing, which requires less energy now. So you have a chance to build back up the energy you need to stay alive, which is why we always hit that point where we can't do another rep or we can't run another lap. And we have to catch our breath, if you will. Now, through the process of slowly training yourself to a higher degree, you're able to become more efficient at using the oxygen you have to create ATP. And so maybe the first time you tried to do 10 push-ups was really hard, but the more you kept putting yourself in a position to require a certain amount of energy, your body gets more efficient at keeping up with you. So you can do 10 push-ups without getting tired. And then you can do 20, then you can do 30, but it's a process of building up to that point. And that's what lactic acid does. When we don't have enough oxygen, which is why we start breathing heavier when we're working out, is we need more oxygen to help make ATP. Our body produces lactic acid to slow us down. Now, this is also where you're going to find something like, let's say, yeast. For all the people that, were making bread at home during our quarantine, yeast in a low O2 environment is going to create not lactic acid, but it's going to create alcohol. And this is a process known as fermentation. So other organisms will go through this process in a low O2 environment. This is called anaerobic respiration. And this is how we use yeast and fermentation to create different types of breads or even actual alcohols, some for consumption and some for fuel sources. This anaerobic respiration, if you will. All right. Now, essentially, that is cellular respiration. That is ATP production within the mitochondria. At any point, feel free to rewatch this video, pause it when you need to, and make sure that you are able to kind of walk through this process of eating food to create energy. Remember, we started here at the beginning. I also included an attachment in Google Classroom with another video that starts at the beginning and works its ways down by someone else, not by me. And hopefully you found this video useful, and I will see you on the next one. Have a great day.